With the increasing legalization and subsequent production and consumption of marijuana in the United States and Canada, the need for proper marijuana education is at an all-time high. And so I thought, what better time to make a comprehensive review of marijuana in the brain. In this guide, I'm going to be discussing the effects that marijuana has on the brain in a way that hopefully anybody with, a high school level education in biology and chemistry will be able to understand. First, I'm going to go over some pharmacology and neurobiology. When you consume marijuana, the chemicals in it will enter your brain and act on a complex receptor system, which changes an even more complex system of brain areas that will eventually change and affect your perception and your behavior. And so I'll outline all of that so we can understand how marijuana produces the effects that it does. After that, I'll be going over a series of important topics relevant to public marijuana use. I'll cover the dangers of adolescent and prenatal use, that is, when pregnant mothers use marijuana and how it affects the brains of developing babies. Then I'll go over what is probably the question of most concern to people, which is chronic use. What happens when you consume marijuana every day, multiple times a day, maybe for years on end? Are there permanent changes in brain structure and function, or maybe even brain damage? Within that context, I'll answer questions about cognition, learning and memory, and motor impairment, specifically relevant to driving laws. Finally, I'll cover a contentious issue, which is marijuana's role in mental illness, both in its ability to contribute as well as treat it. Most of the work I'm going to be using for this presentation is from the scientific literature and has been published in roughly the last 10 years. It's going to be fairly long, but that's the point of a complete guide. Also, before we get started, this is going to be a complete but incomplete guide. Thousands of scientists study the substance around the world and publish hundreds of new papers every month. In the course of making this video, I've probably read maybe 200 or 300 papers, but of course, I'm only one man, and so this review is going to be as complete as I can manage it. So, I hope you find it useful and educational. Here is my guide to marijuana and the brain. Marijuana is a psychoactive drug taken from the cannabis plant. The source of its psychoactivity is from chemical compounds called cannabinoids. Creating a detailed pharmacology of marijuana is pretty difficult because it's made up of about 60 or so, some people estimate over 100, cannabinoid compounds. But the primary psychoactive one, and the one we'll focus on because it's the one that most of all the research has been done on, is delta-9 tetrahydrocannabinol, or otherwise known as THC. Another major one is cannabidiol. These two are purported to make up most of marijuana's psychological and health-related effects. When consumed, these cannabinoid molecules enter the brain and take advantage of a complex system of receptors called the endocannabinoid system. Endo meaning inside, and cannabinoid meaning, the cannabinoid molecules. Endocannabinoids are molecules already present in the brain that bind to these cannabinoid receptors, or the endocannabinoid receptors. And the cannabinoids in marijuana just so happen to make use of these same receptors, which is why they're called that, because they're the same thing. The two main endocannabinoids in your brain are anandamide and 2-arachidonyl glycerol. There are others, like virotamine and nalodine, but they don't really get studied too much. There are also two main types of cannabinoid receptors, which is CB1 and CB2, and roughly speaking, this is how they work. Here we have two neurons. At the connection of the two neurons, we see a typical setup. A presynapse will release a neurotransmitter, maybe serotonin or glutamate. This will act on the postsynapse, where a receptor is located. When the neurotransmitter binds to the receptor, a series of biochemical actions is produced, and some aspect of the neural signal in the postsynapse is altered. Cannabinoids, however, work in the opposite direction, going from the postsynapse to the presynapse, and are so-called retrograde neurotransmitters. Why might a molecule move backwards instead of forwards, and what role do endocannabinoids play in neural function? Well, one critical component of any biological process is a feedback loop. Cannabinoids are thought to modulate the release of other neurotransmitters. When neurotransmitters activate receptors, one of the many results of this activation is the synthesis and production of the endocannabinoids anandamide and 2-AG, which is short for what we covered before. This will move in the opposite direction and attach to either the CB1 or the CB2 receptors in the presynapse. And once it does this, it will produce a series of interesting effects. Activation of CB1 receptor has been found to lead to a reduction in cyclic AMP, increase MAP kinase, and reduce calcium influx. Now, I don't want to spend too much time on all these individual molecules and the molecular processes involved, but basically, cyclic AMP, or CAMP as everyone calls it, is a signaling molecule in cells which does a whole bunch of really important things. MAP kinase is a protein kinase that has a whole bunch of functions and regulates cellular activity. And calcium is one of the most important molecules in neurons, as it's a signaling molecule again, but for many different processes, but most notably for this conversation, is critical for neurotransmitter release. Because of this, CB1 receptors are thought to be presynaptic inhibitors. The collection of biomolecular events caused by CB1 activation leads to less neurotransmitter being released and a decrease in neural activity. They inhibit the function of the presynapse, so a presynaptic inhibitor. Examples of CB1 activation have been shown to reduce glutamate, acetylcholine, and noradrenaline. We can also begin to see how retrograde signaling acts as a feedback mechanism, like the feedback loop. Calcium activates neurotransmitter release. Neurotransmitters bind to receptors. This causes the synthesis of the endocannabinoids. The endocannabinoids move back, activate their own receptors that prevent the calcium release, which is what caused the neurotransmitter release in the first place. CB1 receptors are not just in the brain, but in a number of peripheral organs, and it's one of the most commonly expressed receptors in the brain as well. CB2 receptors are located mostly on immune cells, and so much of the research done on them has been with relevance to the immune system. However, in the brain, they have been shown to lead to enhanced hippocampal long-term potentiation, neuroprotection, neuromodulation, induced cell death, and inhibit tumor growth. So, when you consume marijuana, THC acts as a CB receptor agonist, or a molecule that increases cannabinoid-related activity. So whatever the endocannabinoids do in your brain, the exocannabinoids, or the ones from marijuana, will do a lot more of. This is somewhat obvious because the endocannabinoid system is always active in your brain. Anandamide and 2-AG are constantly activating CB1 and CB2 receptors and modulating your brain activity, but you never notice it, and you certainly don't feel like you're on drugs or you're high. When you smoke marijuana, however, THC, and this is somewhat of a loaded term, but THC will hijack the cannabinoid system that's already in place and really amp up the activity. Cannabidiol does somewhat the opposite. It's called an inverse agonist, and some research indicates that it may even decrease the effects of THC. Now, everything I've told you so far is a fairly simplified but good enough description of the system. The cannabinoid system is actually incredibly complicated with a huge number of molecular interactions and effects. Here's a short list of some of the other receptor molecules that endocannabinoids interact with, and some other molecular effects produced by these interactions. To add to the complexity, CB1 receptors are also found in glial cells. You can kind of think of them as being like brain support cells. They're not actually neurons. Alongside this, there's biogenesis, inactivation, various intracellular signaling pathways, like we mentioned before, and an almost endless list of processes involved, which the layperson just doesn't really need to bother with. And so, now that we know a little bit about how the molecules work in the endocannabinoid system in the brain, and the molecules in marijuana that take advantage of that system, the next important question to ask is, where are the CB receptors located in the brain, and what role does this play in the effects that marijuana has? And so, in this part of the video, I'll talk a little bit about how marijuana produces each of its specific effects on specific brain areas. Cannabinoid receptors are one of the most widely prevalent receptors in the brain. However, they're most highly concentrated in the hippocampus, the striatum, the cerebellum, the hypothalamus, certain parts of the neocortex, the thalamus, and the brainstem. Left to its own devices and untampered by drugs, the endocannabinoids in these brain regions play a critical role in anxiety, appetite, mood, pain, depression, and memory. Consuming marijuana changes the level of activity of this complex receptor system, and so produces a distinct set of psychological and behavioral effects, such as short-term memory loss, anxiety and stress, cognitive impairments and reduced focus and attention, mood alterations, enhanced body awareness, discoordination, sleepiness, increased appetite, pain reduction, and of course, the most obvious, feelings of euphoria. The hippocampus is a medial temporal lobe structure, which means that it's near the middle of the piece of the cortex located on the sides or the temples of your brain. Here's where they are in this image I'll show you. The hippocampus is understood as being critical for episodic memory, so memory for episodes or events that happen in your life, spatial navigation and memory, or how you find your way around and where you remember where things are and where to go, temporal ordering, so determining the order in which things happen, and anxiety. How it does all of this is actually pretty complicated. It has a well-defined but pretty complex set of inter and intra connections between itself and other brain regions. And lots of computational models and hierarchical theories of function exist in an attempt to make sense of all that, which we don't need to go over here. Some things that marijuana has been shown to affect in the hippocampus are decreased GABA, decreased glutamate, those are just two examples of neurotransmitters, and impaired long-term potentiation and long-term depression, which I'll go over in a bit. GABA is an inhibitory neurotransmitter that reduces neural activity, and glutamate is an excitatory neurotransmitter, which will increase neural activity in general. When CB receptors are activated, both GABA and glutamate are decreased in interneurons and principal neurons, respectively, which is just what they're called. It's the names of the neurons. And the result of this is an impairment of a special type of neural activity in the hippocampus, which is famous for, called long-term potentiation, and its counterpart, long-term depression. LTP and LTD, as they're shortened to, are thought of as being some of the main mechanisms by which the brain learns new information. It involves physical changes to the structure and connection of neurons with each other. When high frequency bursts of activity happen, the neural connections strengthen, and as an outcome, a stronger signal or a potentiated signal occurs. If neurotransmitter release is decreased, then it's a lot harder to get LTP. And with no LTP, there's typically no learning. Behaviorally, alterations in hippocampal function affect memory and navigation. Although marijuana's effect on memory is one of the most talked about anecdotally, the scientific evidence for this is abundant and relatively straightforward. There are learning and memory tests that you can give to laboratory animals like rats and mice, and if you give the animals marijuana, they'll have significant learning impairments in these tasks. Interestingly, if you give animals drugs that are cannabinoid receptor antagonists, and remember that marijuana is a CB receptor agonist, if you give them CB receptor antagonists, you can see learning improvements in these tasks. It's thought that marijuana's effect on LTP and LTD and neurotransmitter reduction in the hippocampus, as well as many other brain areas that form memory-relevant circuits, like the amygdala and the prefrontal cortex, is what's primarily responsible for its effects on memory impairment. It's important to note that memory is not really a uniform thing, and that there's multiple different types of memory that are thought to rely on multiple different types of brain areas, and marijuana can impair some, but at the same time improve others. Specifically, fear and emotional related memories in the amygdala. It reduces glutamate and GABA release, but just like it does in the hippocampus, but when this happens here in the amygdala, it results in memory improvements for some interesting reasons. Specifically relating to a kind of active forgetting process called memory extinction, but we'll talk about that much later when we go over PTSD. One way in which THC and marijuana is thought to contribute to anxiety is through neuromodulation of the amygdala. Parts of the amygdala belong to a sensory processing circuit that goes from the cortex, where visual images are processed, through the amygdala, where emotional associations are learned and attached to events and objects, and down to the hypothalamus, eventually triggering the HPA axis, the hypothalamic pituitary adrenal axis, which is where hormones are produced. In this case, stress hormone production. We can think of this circuit as a kind of fear sensor and fear learner, picking up on potentially dangerous things in our environment and eventually activating a response of fear and anxiety to them. Well, if you alter neurotransmitter function in this pathway, making it more or less excitable, so if it's more excitable, well then it gets set off all the time by every little thing, making everything stressful and scary. But if it's less excitable, then nothing is scary and you can relax more. The fact that the amygdala is comprised of many substructures and receives input from other structures that are also affected by marijuana makes outlining the relationship between marijuana and anxiety kind of difficult. Marijuana produces what's usually referred to as a U-shaped curve when it comes to anxiety. For example, if you block CB1 receptors in the brain, you can increase anxiety. And if you activate CB receptors, let's say with marijuana, you can decrease anxiety. But at some point, if you activate them enough, you begin to increase anxiety again. Low doses are good, high doses are bad. Not only this, but the two main cannabinoids we've been talking about, THC and cannabidiol, produce somewhat opposite effects in the brain. Cannabidiol and THC are, of course, not the same molecule, and they act on CB receptors somewhat differently. They exist in varying degrees and ratios in different types of marijuana, and so one possibility for marijuana's U-shape relationship to anxiety may be the differing degrees of effect that each molecule has on the nervous system depending on how much of each you take. Cannabidiol is thought to produce anxiety-reducing effects acting on CB2 receptors in the limbic and paralimbic brain regions. One of the most common described effects of marijuana is the munchies, or an increase in appetite and an increase in the subjective quality of food taste. Circuitry relating to eating has been well documented and outlined extensively and covers everything from subjective taste, satiation, energy balance, and the rewarding and emotional aspects of food, and distinct problems in the circuitry relating to obesity and eating disorders. Much of the eating circuitry is covered with cannabinoid receptors, and so we'll cover a little bit of that right now. The nucleus accumbens belongs to a much larger set of brain regions called the basal ganglia that collectively control things like motor learning, sometimes popularly called muscle memory, the formation of habits, motivation, dopamine release, and its production relating to pleasurable experiences and reward. The nucleus accumbens is sometimes famously called the reward center of the brain, if not slightly inaccurately because reward circuitry is complex and involves a whole bunch of structures. The nucleus accumbens is loaded with CB receptors. An activation of those CB receptors in the nucleus accumbens modulates dopamine release and the activity relating to reward. And, just like cocaine or sex, food has rewarding properties as well. If you inject anandamide into the nucleus accumbens of rats, you can increase their eating behaviors. Unlike its effects on glutamate and GABA, THC will increase dopamine release in the nucleus accumbens, and this also coincides with a behavioral increase in eating behaviors. Lots of evidence from animal work has been done comparing the palatability of various foods and how food preferences change with THC use. So, literally studies of rats getting stoned and seeing what foods they like. And this has indicated that it's not just an increase in appetite, but also changes in taste. Exactly how marijuana does this is still unknown, although some evidence indicates that it's partially controlled by distinct parts of the nucleus accumbens as well. It's also important to note that all of this is from animal studies, and that marijuana's effects on dopamine in humans has been highly debated, because there's not nearly as much evidence as there is from the animal studies. The nucleus accumbens sends a great deal of signals to another brain region, a very important brain region called the hypothalamus, specifically the lateral, paraventricular, and ventromedial hypothalamus. The names just indicate where they're located, kind of like anatomical geography, so don't worry about it. The hypothalamus is a small but powerful brain region that functions as the main producer and mediator of hormones in the body. The lateral portion has long been studied for its role in eating. It causes the release of orexin, a neuropeptide related to appetite regulation, and melanin concentrating hormone, which has a role in energy homeostasis. Neurons containing orexin and melanin hormone also contain CB receptors, and so activation of the CB receptors by marijuana influences orexin and melanin hormone activity in such a way that results in increases in eating. Much of the evidence for nucleus accumbens involvement in eating applies here as well. Injecting anandamide or THC into the hypothalamus of rodents can increase eating behaviors. Other brain areas located in the brainstem, the most evolutionarily old part of the brain, and usually contains the most basic and important functions for staying alive, like breathing, sleeping, and heart rate, well, they also control eating. Places like the parabrachial nucleus, dorsal motor nucleus of the vagus nerve. The vagus nerve is most famous for, I guess, being responsible for the gag reflex and vomiting, although it's got a number of other really cool functions too. The nucleus tractus solitaris. Unfortunately, some anatomy terms are never really easy. These are also areas subject to a lot of cannabinoid receptor regulation. Finally, modulation of the cannabinoid system modulates the opioid system, which is a whole other distinct molecular system in the brain with separate functions, and the opioid system also affects appetite and eating behaviors. Evidence for this comes from the fact that cannabinoid-related increases in eating behaviors can be stopped with the administration of opioid antagonist like naloxone. THC has been shown to cause the release of beta-endorphin in the nucleus accumbens as well, a related endogenous opiate, kind of like there's endogenous cannabinoids as well. Now, much of the evidence I'm going to talk about for this section comes from brain imaging studies like positron emission tomography and functional magnetic resonance imaging. Don't worry about what all that means, it's just what they're called. Basically, you can get a person to sit into a chair, do some computer task, or watch a video, or in this case, you can give them a drug. And these highly complicated imaging techniques can tell us what parts of the brain become active when they're doing that thing. When you consume marijuana, there is a distinct and noticeable high feeling that is produced. It's called intoxication. And a number of changes have been observed when this happens. The majority of brain imaging studies have found increased global cortical activation directly after consuming marijuana. Basically, this means the entire cortex collectively becomes more active. Everything you see here, all these bumps, these are part of the cortex. As a brief review, the cortex is a thin layer of neural tissue surrounding the brain that is heavily connected both to itself and to the brain areas underneath, so-called subcortical brain areas, like these ones here. It is thought to be important for complex sensory processing, intelligence, and cognitive capacity. Evidence from this comes from the fact that many different pieces of the cortex are related to complex behaviors, like the fusiform gyrus for face recognition or the intraparietal sulcus in mathematical calculations. Also, as you move up the evolutionary tree, the cortex appears in higher order organisms like mammals and gets more complex as you move up to primates, and that its most complexity in humans, or arguably dolphins, but that's a whole other video in and of itself, which we're not going to talk about here. A few studies have found that the right hemisphere, the brain is divided up into two hemispheres, the left and the right, that the right cortical hemisphere is more active than the left during marijuana use, and some scientists have proposed that this might relate to marijuana's effects on creativity and emotional awareness. To be a little bit more regionally specific, heightened activity is most often seen in the frontal lobe, the limbic cortex, the paralimbic cortex, and the cerebellum, which is not part of the cortex. The actual feeling of intoxication or being high is associated with reduced activity in the right parietal regions, seen here, and the anterior cingulate cortex. The anterior cingulate is this very interesting chunk of cortex associated with emotion and decision-making, pain, reward, and some people think it's quite critical for consciousness. The right frontal lobe is also found to be consistently activated during marijuana use, and it's here that a lot of the more interesting subjective effects are proposed to take place. Things like the feeling of intoxication and a disturbed sense of time. On a side note, it's not a cortical structure but relevant to the effects we're talking about. The cerebellum is this large cauliflower-looking structure underneath the brain in the back, and it's loaded with cannabinoid receptors. Interestingly, the cerebellum has long been implicated in time perception, and some researchers have shown that cerebellar activity under marijuana is negatively correlated with accurate time perception, which would make sense that a structure involved in the timing of events is less active when you can't accurately perceive time, which is a commonly talked about effect of marijuana. Marijuana has been shown to affect a number of important cognitive functions in people. These are verbal working memory, psychomotor function, attention, inhibition, and time estimation. Much of these behaviors rely on the prefrontal cortex, an area that is, you guessed it, even farther in front of the frontal cortex, and its relationship to a variety of these other areas, which we've already talked about, like the hippocampus, the basal ganglia, the amygdala, and of course other cortical regions. Now, a detailed neurobiology of the neural networks behind all of these behaviors is way beyond the scope of this video, although I will mention a few important details about how marijuana affects prefrontal cortex function. Dopamine, which we've talked about already, is produced in the ventral tegmental area, shortened usually to the VTA. This is a small area in your brain stem, and it creates all the dopamine in your brain and sends it out to other places to use. And one of the places it sends it to is the prefrontal cortex, where it controls glutamate and GABA activity. THC in marijuana plays around with all these molecules and so alters prefrontal cortex activity. We talked about the hippocampus earlier in reference to long-term memory. Well, the prefrontal cortex has an important role in short-term memory, sometimes called working memory, keeping things online for, seconds at a time. Of course, if you have problems remembering things for any longer than a couple seconds, it's going to be really hard to pay attention or perform cognitively complex tasks. Now, what I've just described so far are the various brain areas and the neural functions that marijuana affects and how that relates to the primary reported effects of marijuana. Changes in behavior and perception. These are some of the most widely studied effects, however. There's a massive list of effects that marijuana has on the brain, and here's just a short list of a few. Like I said at the start, it's a very large field of study. Also, everything I've mentioned so far was in relation to marijuana's acute or short-term immediate effects. However, many people are far more interested in its chronic effects, specifically whether chronic use has any potential long-term impairments in brain function, and whether or not these impairments are permanent. Chronic use of marijuana has been shown to downregulate CB1 receptors in the brain. Downregulation is when some process in a cell is decreased and happens less over time. This is a very common and widely known effect in chronic drug use of almost any kind, not just marijuana. Because there is higher than normal levels of activity for long periods of time, the body compensates by reducing the production of molecules involved. And this downregulation of CB1 receptors after chronic use is assumed why habituation occurs, where you need more of the drug to get the same effect. While it might suck that you have to smoke twice as much to get the same high feeling, your brain's endocannabinoid system is now greatly reduced, even when you aren't high. And so all the things I've just been talking about that the endocannabinoid system regulates, like memory and anxiety, pain, mood, and cognition, will be affected. This is why there are lasting residual effects of chronic drug use. It takes a while for things to go back to normal. And this brings up one of the most widely asked and controversial topics about marijuana, and that's whether or not the chronic effects on cognition and behavior are permanent. And that's what we'll cover right now. The effects of chronic long-term marijuana on brain structure and function are uncertain, as very little reliable evidence has been shown to consistently lead always to significant impairments in brain structure, function, and behavior. Just when one fMRI study comes out showing long-term reductions in the hippocampus size or reduced overall brain size, another one will come out showing no effects at all. This type of research is also plagued by a number of methodological issues as well. It's very difficult to find a consistent group of participants for studies who have all smoked the exact same amount across an equal amount of time over their lifetime and have quit for the same period of time as well, all within the same study, as well as between and across studies to make comparisons. It's also quite difficult to determine if the brain and behavioral differences found were due to chronic drug use or if the brain and behavioral differences were already present in the people who then decided to do drugs long term. In other words, it's not clear if chronic marijuana use leads to, let's say, a smaller hippocampus, or if people who have a smaller hippocampus are more likely to do drugs chronically. Either way, the question has stimulated a lot of research. Although there's no consensus, some effects have been found many times over many studies. These are reduced hippocampal size, reduced prefrontal cortex size, and reduced cerebellum size. Other areas which have been shown to be affected somewhat inconsistently or inconclusively are the amygdala, precentral, fusiform, and cingulate gyrus areas in the cortex, whole brain or entire brain volume, white matter density, the insula, and general brain connectivity. All of these brain areas, of course, are loaded with CB receptors, so it's not surprising that when damage is seen, it happens most commonly in those parts. All of these size reductions are usually positively correlated with heavier use, meaning that the more you use, the more likely damage will be seen. How potential damage might come about could happen in a number of different ways. Marijuana alters neurotransmitter activity. Altered neurotransmitters will lead to changes in how signals get sent across the brain, and so if this lasts for long enough, it might slowly atrophy the affected brain areas. The cannabinoid system also regulates stress hormones via the HPA axis, like we talked about. And so dysregulation of the stress system can cause brain damage in the long run. Most importantly, many studies have observed a reduction in the hippocampus, prefrontal cortex, and cerebellum, and that they don't go back to normal after extended periods of abstinence. This is especially true for the most heavy users of marijuana who use every day, multiple times a day, usually up to several years, as well as for people who use prominently in adolescence. These are some of the structural or physical damages that might occur, but now let's talk about brain activity that's associated with behaviors. Rather than spending a lot of time detailing all the various studies, I've made a chart with data from a review paper on acute versus long-term effects of marijuana on cognitive function. It was done by Rebecca Crane, Natanya Crane, and Barbara J. Mason, who are part of the Committee on the Neurobiology of Addictive Disorders at the Scripps Research Institute, to give credit where credit is due. This review paper was published in the Journal of Addiction Medicine in 2012. It's public access, so go check it out if you want to. Functional brain imaging can be a little tricky to interpret because it's not necessarily the case that if you have more brain activation during some behavior, that means that you're doing better or that your brain is performing better or that it's healthier. For example, if you perform very poorly on an attention test or some decision-making test or some cognitive task, which chronic marijuana users have been shown to do, but also show increased brain activation in the areas that are thought to be responsible for those attention or cognitive-like behaviors relative to non-marijuana using subjects, then that means that your brain is trying harder, putting in more effort, and getting less performance to show for it. And that's not usually a good thing. And it's exactly this type of brain function that we see in a lot of fMRI studies of chronic marijuana users. The findings on chronic marijuana users' brain function during cognitive tasks usually takes two types. Either there will be an increase in brain activity in an important brain structure with no difference in cognitive performance, although sometimes there's reduced performance which is observed, or there'll be decreased activity in the important brain regions in question and an increase in activity somewhere else in the brain, with also no difference and sometimes a reduction in the cognitive performance. This can mean one of two things. It means that either your brain is working harder to achieve the same results, or that your brain is making use of extra brain regions because of a lack of functionality of the primary brain regions in order to achieve equal performance, something called compensatory recruitment. Basically, in a healthy brain, areas X, Y, and Z will be needed to perform some task. But after years of heavy marijuana use, X, Y, and Z don't work as well, and so areas A, B, and C have to kick in to help out. You still perform just as well, but at the cost of efficiency. In summary, chronic marijuana use may have permanent damaging effects on brain structure, function, and behavior. Although the research is inconclusive, with many researchers still performing studies to this day and finding no effects, it appears that when effects are observed, it is more often seen and the deficits are greater in the heaviest users, which of course is very ill-defined, not well-defined, and in people who begin their chronic use earlier in adolescence, which is what we'll turn to now. The reason why studying adolescent marijuana use is so important is because, one, the brain of a young person is not the same as the brain of an adult, and so there are many parts of the brain that are still developing well up into people's early to mid-20s, like the prefrontal cortex. And two, this is the period of time when most people actually first come into contact with the drug and they first start using it. So if you smoke marijuana when you're between the ages of, let's say, 10 and 19, how does this affect your brain development well into adulthood? Well, heavy marijuana use during adolescence is associated with many of the things that I've already talked about. Impairments in memory, attention, cognition, except that the outcomes seem to be much worse, with impairments lasting well into periods of abstinence, indicating that they might be permanent. This is particularly true when considering the fact that the prefrontal cortex is part of the brain that takes a very long time to develop and may not be finished until we reach the age of 25, and that it's also loaded with cannabinoid receptors. Things get much worse the more you use and the younger you start using. And I'll quickly go over some of that evidence now, which there's actually quite a bit of. Early regular use of marijuana in adolescence has been associated with impairments in attention, memory, self-control or inhibition, decision-making, visual search, risky sexual behavior, and a permanent reduced overall IQ. So here's some papers supporting that, and here's a bunch more, and here's even more. As we can see, the data on the negative impact of regular chronic marijuana use during adolescence is a lot more straightforward than it is in adults, with the consensus being much stronger that there's a greater risk of doing your brain some serious damage if you use marijuana quite heavily. A somewhat related topic is what happens when your mother uses marijuana when you're in the womb. This is a period of time called prenatal, before you're born, and represents a particularly sensitive developmental period when you're susceptible to all sorts of negative effects from toxins and drugs and stress. Many of the same things I've already talked about hold true for this as well, lasting impairments in cognitive function and impaired brain development. So that's the discussion on the potentially permanent effects of chronic drug use. Now we'll move into some other hopefully interesting topics. And I like this one particularly because it's something that I worked on in my undergrad. Gateway drug theory, sometimes called stepping stone theory or the escalation hypothesis, is the idea that the use of one psychoactive substance can lead a person to using other psychoactive substances over time. However, the explanations for why can be a little bit confusing. The escalation usually goes something like this. If I take drug X for 1 year, after a while, drug X just won't do it for me anymore and it won't really get me high. And so, in order to get high, I have to upgrade to some other, stronger drug. Another way it might work is that if I take drug X for some period of time, it permanently changes my brain in such a way, typically in the reward system like we talked about earlier, so that if I were to take a new drug later on, my risk of becoming addicted to that new drug would be increased. One drug in particular, marijuana, has been claimed to be a gateway drug for many, many years, and not without reason. There do exist links between marijuana use and subsequent use of other illicit drugs. When studying large human populations, so this is epidemiology, the percentage of people who use marijuana who go on to use other drugs is higher than the population of people who never use drugs in the first place. And there is evidence, actually, that marijuana use can alter the brain to make it respond differently to other types of drugs, like opiates and cocaine, that people with no prior use just won't have. In other words, past marijuana use can prime or set up your brain to make it more susceptible to addiction to other drugs. Mind you, this evidence is in rats and is by no means agreed upon, nor is there a consensus in the literature because it just hasn't been studied that much. Now, with this type of evidence, it might seem clear that marijuana is probably a gateway drug and can lead to other future drug problems. However, there's a reason why gateway drug theory is so heavily debated and contested, and some people claim that it's not even real. It is, in fact, quite nuanced, and by no means is as simple as the drug changes in your brain lead you to take more drugs, or this drug just doesn't do it for me anymore. The idea that you could take a drug and it won't do it for you anymore really only works when you're working within the same class of drugs. Like if you take a mild opiate and the mild opiate doesn't work, so you upgrade to a higher opiate. But of course, there's different classes of drugs for a reason, and they all work differently on the brain. For instance, a particular type of person takes drugs to begin with, and so there's an inherent bias in the types of people who are likely to take more drugs after their first drug. And so a genuine study of the cause and effect can't be done, because you can't ethically just take a bunch of people who have no interest in drugs, give half of them marijuana for a couple months, and then track the rest of their lives. Drugs also don't exist in isolation. They exist more or less in different groups of people, varying according to the region. It would be hard to imagine marijuana as a gateway drug if it was the only drug available where you lived. No one would have the chance to move on to anything stronger. Another explanation for the gateway drug theory is the idea that if you're interested in taking drugs, you take a drug and it works out quite nicely, and then you might be inclined to try out other drugs because your first experience went so over well, which is perfectly reasonable to assume. Some people do move on to try out other substances which have a higher risk of addiction, like opiates and amphetamines and cocaine. However, most people don't. There is some evidence, at least in rats, to show that heavy use of marijuana can change how the brain responds to other drugs. And so gateway drug theory isn't completely false, which is why more recent models have attempted to make sense of multiple factors that contribute to people using stronger and potentially more dangerous substances, accounting for environment, upbringing, genetics, and drug use patterns. It's also very commonly stated that marijuana is not an addictive substance, and while it's certainly true that when compared to other substances of abuse like nicotine, heroin, or amphetamine, that its addictive potential is quite low. The withdrawal symptoms are usually quite mild, and some even arguing that marijuana addiction isn't real or impossible or is just made up. Regardless, this doesn't restrict us from talking about marijuana's rewarding and pleasurable properties, which might lead people to do it all the time. Cue-related craving is a phenomena where various things that become associated with drug use turn into cues or triggers that increase people's cravings for the drug that the person is used to using. And it's one of the kind of foundational aspects of addiction. So, for example, seeing pictures of the devices that you use to take drugs, like a pipe or a cigarette, seeing the drug itself, hearing words or phrases about the drug, has been shown to increase people's self-reported drug cravings. This has been shown with substances like heroin, cocaine, alcohol, and there's some evidence that the same process, relying on the same brain areas as these other drugs, happens in frequent marijuana users. In an imaging study using functional magnetic resonance imaging, when presented with an image of a pipe, frequent marijuana users show an increased activation in the VTA, the dorsal anterior cingulate cortex, the amygdala, the inferior frontal gyrus and the insula, the central gyrus, the cerebellum, and the thalamus. And you can see on here all of the various behaviors and perceptual processes that are associated with those brain areas. A few other studies have found similar results. One thing that almost everybody is aware of is the dangers of driving vehicles while drunk. And so there are strict laws against drunk driving, as well as being impaired by other substances that may impair your ability to drive. Impair. The topic of driving under the influence of marijuana has come up a lot more recently due to its increasing use and legalization, with a common claim among users that driving while high is not dangerous or even safe due to the increased paranoia, and so you drive slower and you drive more carefully. Well, some work has been done on this as well, both epidemiological, examining call reports, and others experimental, using subjects on marijuana and driving simulator tests. Since we'll be talking quite a bit from now on about epidemiology, I'll define what it is. It's the study of health outcomes in populations and an attempt to make causal relationships between things that people do or things that happen to people and their overall changes in health. Proving marijuana's effect in car accidents is actually pretty difficult. For one, marijuana has a very short half-life in the body, meaning that it breaks down very quickly. And so by the time you've been arrested and then tested for it, chances are the levels of THC in your blood cannot be distinguished from what the levels would have been if you'd smoked yesterday or in the morning or hours and hours before you got arrested. Not only this, but marijuana is also not the only drug involved in drug-related car crashes, making the causality even trickier. The epidemiological data points to a couple things. First, regular marijuana use is associated with an increased risk in motor vehicle accidents, and the greater the concentration of THC in the blood is associated with higher motor vehicle accident culpability, meaning that when an accident happens, the higher you are, the greater likelihood that the accident was your fault. So here's a small list of some studies that have examined this if you're interested in checking them out. Now on to experiments. When participants in a study are given marijuana and then put into a driving simulator, some common results tend to appear across studies. Probably the most commonly reported effect is an increased reaction time, which is bad when you're driving. There are also increases in lane weaving and driving outside of defined borders, equally bad when you're driving. However, compensation is also very common, with the average speed being a lot slower. Impairments are also the greatest within the first hour after smoking, and they usually tend to decrease after that as the drug effects fade away. Because driving is a complicated task filled with all sorts of subtasks like paying attention to multiple moving objects, tracking the rates of measures of things like speed and gas, following learned rules of the road, spatial navigation, and all of these things happen all at once in real time. Given what we know about marijuana's effects on cognition, it's really no surprise that it would impair such a cognitively demanding task like driving. Finally, I'm going to end this video off talking about marijuana's relationship with mental illness, both how it contributes to it and how it helps to alleviate it. The relationship between marijuana and schizophrenia has been often debated. Schizophrenia is a complex mental disorder characterized by abnormal social behavior and difficulty determining what is real and what is not. Common symptoms are things like hearing voices, difficulty in comprehension, and a general lack of motivation. First, I'll cover some evidence for this potential relationship and then talk a little bit about what it might mean, because it's certainly not as simple as marijuana causes schizophrenia. First off, like epidemiology, longitudinal studies have found that cannabis use is associated with an increased risk in schizophrenia. So here's a bunch of work showing that. Although this isn't always the case, so here are some important counterexamples. Of course, there's always the possibility that people with an emerging mental illness might seek out drugs of abuse in order to cope, and that the drug use can then intensify the development of the already almost present mental illness. This is a constant problem with a lot of epidemiology work and longitudinal studies of marijuana use in schizophrenia. An association is almost always found, but the direction of the association is almost always uncertain. Here are some of the more specific negative effects of marijuana as they relate to the symptoms of schizophrenia. Suspiciousness, paranoia, fragmented thinking, disorganized thought, altered perception, derealization, and depersonalization. Many of these are the positive symptoms of schizophrenia. It should be noted that these effects have been extensively researched with regards to THC specifically, but also CBD, cannabidiol, which oftentimes shows antipsychotic, protective effects, the opposite of THC. Of course, these effects only occur while the person is on the drug, right? And disappear soon afterwards, and so are usually referred to as being psychomimetic, or mimicking psychosis. Now, if all marijuana did was, temporarily mimic the symptoms of schizophrenia in a small subset of users, well, that would, that'd be fine. Who would care, right? Because it wouldn't last, and you'd be fine when you're done. But what about the idea that marijuana may induce lasting or even permanent psychosis? Indeed, this is a commonly reported phenomenon that marijuana use has triggered or initiated psychotic or schizophrenic symptoms in some people. Now, the fact is that most people who smoke marijuana don't experience psychosis and don't get schizophrenia, and so a direct causal link should not be made. But instead, the idea that there exists a group of people who have a complex biological and environmental disposition to psychotic illness, and that marijuana plays a critical role in the development of that illness, not on its own, but only when these other factors have an important role. Things like genetics, child abuse, family history. Marijuana is, for many people, the thing that sets their schizophrenia into motion. But these were people who were halfway there, if that's the way you want to phrase it. And some other powerful psychoactive drug may have done the same thing. For example, amphetamine-induced psychosis is also a described phenomenon. To sum up, the link between marijuana and psychosis might look something like this. Within most people of the population, marijuana will not produce any serious negative or permanent effects. However, in people who have the unlucky combination of a genetic predisposition to psychotic-like symptoms, poor childhood upbringing, and a family history of mental illness, marijuana may produce what is referred to as cannabis-induced psychosis. This may be temporary, as long as marijuana use stops. However, in about 50% of cases, full-blown schizophrenia is developed. This is why marijuana is thought to contribute to roughly 8 to 14% of all schizophrenia cases. The relationship between marijuana and anxiety is a tricky one that we've touched on a little bit already. In small quantities, marijuana has been shown to reduce anxiety, but in large quantities it's known to produce anxiety. This is mostly related to THC. While one of marijuana's other primary cannabinoids, cannabidiol, is the one that reduces anxiety. It's this knowledge that has led to some special strains of cannabidiol-heavy marijuana to be developed and used in treating anxiety disorders. However, these are, after all, special strains that have very specific medical purposes, and so we're left with the question, can regular marijuana use lead to the development of an anxiety disorder? Once again, epidemiology work has shown that marijuana use and anxiety disorders are highly correlated. However, just like before, proving causality is difficult because people with high levels of anxiety also use marijuana therapeutically. It appears that marijuana and anxiety go hand in hand, both to reduce it but also to contribute to it, depending on the situation, how much it is used, and the person using it. So, a bit tricky. In a meta-analysis of the association between marijuana use and depression, Lev Ran et al. found a modest increase in the risk for developing depression in those who use marijuana. A meta-analysis is a type of literature review comparing sets of similar studies. Several longitudinal studies have also found a risk for developing depression in people who frequently use marijuana. Now, why marijuana use might lead to depression is confusing because just like with anxiety, marijuana use can also be used to treat depression. Considering that cannabinoid antagonists like rimonabant, remember that THC is an agonist, these can cause depression. Marijuana has the ability to alter emotional experiences, but how in relation to depression is a little unclear. Acute marijuana use tends to be very antidepressive, since one of its main effects is euphoria. Indeed, cannabinoid receptor activation has been shown to lead to an increase in serotonin activity in the prefrontal cortex, increased neurogenesis in the hippocampus, and reduced HPA axis stress activity, all of which are associated with antidepressant effects. Some have speculated that marijuana's ability to lead to depression in the long run is not actually neurobiological in cause, but instead that long-term heavy use leads to lower life success and things like less education and unemployment, which on average is true given the epidemiology that we've looked at, which of course could then lead to depression. So more of an indirect cause. So just with anxiety, short-term doses seem to be beneficial, long-term high doses can be harmful. Post-traumatic stress disorder is an anxiety disorder that develops after someone has experienced a traumatic, emotional, intense, or violent event, usually while the person in question is unable to do anything about that event. Common symptoms include heightened central nervous system arousal, sleep disturbances, nightmares, depression, anxiety, and avoidance behavior, and emotional numbing, and traumatic event flashbacks. A neuroscience of PTSD, if that's what we want to call it, has been quite well developed, with studies showing that the hippocampus, the amygdala, and the anterior cingulate cortex being primarily affected. Together, these three brain areas make up an important part of the fear learning and emotional regulation system. In general, amygdala activity heightens while hippocampal and cingulate regions are reduced. This is relevant as the amygdala, like we mentioned earlier, is very important in learning and expressing fear, while the hippocampus plays a critical role in regulating the stress response. So more amygdala, less hippocampus means more fear and more stress. Marijuana is a common coping tool by many people who have PTSD, and its main purpose being to aid with sleep, reduce stress, and reduce anxiety. Some of this should be fairly obvious to you now, since we've already covered anxiety, and marijuana is frequently described by those who take it as increasing sleepiness and reducing overall arousal. Activating the hippocampus via cannabinoids also appears to be beneficial in this group, as they have reduced hippocampal activity. However, one effect of marijuana is particularly interesting with regards to PTSD. But a little bit of an anatomy lesson first. The ventromedial prefrontal cortex sends reciprocal, or back and forth, connections with the amygdala. The amygdala is also heavily connected to the hippocampus, which is the place of memory storage. It is thought that when stimuli from the outside world activate the amygdala, the ventromedial prefrontal cortex inhibits or blocks the flow of information to the hippocampus and prevents the recall or bringing up of memories. In PTSD, this ability of the ventromedial prefrontal cortex to do its job is reduced, leading to more memory recall more often. And with a hyperactive amygdala primed for fear, this means more uncontrolled traumatic flashbacks. As I mentioned in the section about memory, one of the effects of marijuana on the amygdala is enhanced extinction memory. Extinction is a kind of unlearning. It's not forgetting exactly. Slowly, the things that are associated with one another, in this case, a trigger is associated with a memory of the traumatic event and leads to re-experiencing it, or the recall of the memory. These things become unassociated over time, as the two things just don't happen, paired together over time anymore. Marijuana has been shown to enhance extinction learning in rats and in mice and in humans. It's because of this that it offers therapeutic potential in helping people who suffer through PTSD to learn to forget about the traumatic event. Now this video is pretty long already, so I'll cover a couple more things quite briefly. Marijuana has been shown to be a viable treatment for pain. CB2 receptors have been tried as a target for pharmacological action against both acute and chronic pain, and for neuropathic pain as well, resulting from nerve damage. This is pretty useful, considering that all the psychoactive effects of cannabinoid receptor activity are thought to come from CB1 receptors, meaning that there's a potential for a cannabinoid-based pain medication that doesn't produce much of a high. One proposed mechanism by which activation of the CB2 receptor is proposed to suppress pain is through its anti-inflammatory effects. Inflammation irritates nociceptors, or the receptors in your body that send pain signals, and activation of CB2 receptors can reduce inflammation and, as a result, less nociceptor sensitivity and activity. CB2 receptor activation also leads to an increase in beta-endorphin production, an endogenous opioid. Most people are quite familiar with the term endorphin, a chemical that acts on the opioid receptors in the brain and the body to reduce stress and reduce pain. Considering marijuana's potent effects on memory processes, some have questioned whether a link might be made between using the drug long-term and the development of Alzheimer's disease. As we have covered previously, lower hippocampal volumes were observed in some people who were heavy long-term smokers. However, this is only one side of the coin. The potentially memory-damaging aspect of this drug, when used heavily, are related to CB1 receptor activity, while the CB2 receptor is widely noted for its anti-inflammatory and neuroprotective effects, which is why some scientists have studied marijuana as a therapeutic substance for the treatment of Alzheimer's disease, as opposed to a causative one. It has been shown to prevent amyloid plaques from building up, slow down neurodegeneration, enhance neurogenesis, the birth of new neurons, and reduce brain inflammation. Similar effects of marijuana have been found for multiple sclerosis and traumatic brain injury as well. So there it is, my complete yet still incomplete guide to marijuana in the brain. As of now, the knowledge of this drug and how it affects brain function, behavior, and mental illness is vast. However, many areas still need considerable work. Much of the epidemiology literature is flawed in many serious ways, like the inability to control for confounding factors like the influence of other drugs and that people use, like alcohol, amphetamines, and opiates. There's also a lack of consistency in how much marijuana is used by participants and for how long for a lot of these studies. Although by no fault of their own, since these are just the inherent limitations of epidemiology work, and epidemiology is of course quite useful in its own right. There's also discrepancies between the animal and the human literature. For example, THC has been shown to reliably alter dopamine function in rats and mice, but its effect on dopamine in human brains is quite limited. With that being said, this video is timestamped for 2017 because who knows what else we might discover about marijuana in the future. If you found any glaring mistakes or something important that I might have missed, please leave a comment below. And as always, thank you for watching and have a great day.